So today we'll be going through the clinical pharmacology and therapeutics related to hyperlipidemia, atherosclerosis, and downstream cardiovascular disease. So first we're going to go through a brief overview of cholesterol metabolism and transport and how this is altered in hyperlipidemia. And then we'll discuss how the indicated therapeutics will augment this disease mechanism. So let's start with cholesterol metabolism and transport. So there are two primary pathways in the body that are responsible for this process. And the two pathways are the exogenous pathway, and associate this with dietary cholesterol, and the endogenous pathway, and associate this with de novo liver synthesis of cholesterol. So starting with this exogenous pathway. So dietary fats like cholesterol and triglycerides are absorbed from the small intestines, but because they are water insoluble, these have to be packaged in the form of lipoproteins. So in these intestinal epithelial cells, cholesterol and triglyceride are combined with phospholipids and apoproteins, ultimately forming a chylomicron. And now these chylomicrons have a very hydrophobic core that's made up of these triglycerides, which are shown in blue, and cholesterol, which is shown in red. And this very hydrophobic core is then covered with this hydrophilic exterior, again, of those proteins and phospholipids. So chylomicrons can then enter the circulation once they're formed and interact with lipoprotein lipase, or LPL. And this occurs in the capillary lumens. LPL actually acts to hydrolyze triglycerides to free fatty acids to then be utilized by the surrounding adipose tissue and muscle tissue. This triglyceride hydrolysis actually shifts the content of these chylomicrons from being very triglyceride rich to being very cholesterol rich, which I'm showing you here. These particles are now called the chylomicron remnants, or just remnants, and these remnants continue in the circulation until they reach the liver, where they are taken up after interacting with their remnant receptor. And this basically just delivers dietary cholesterol to the liver, which can now be used to participate in steroid synthesis, cell membrane formation. These cholesterol molecules can be repackaged into other lipoproteins, and we'll talk about this a little bit more with the endogenous pathway. And this cholesterol can also be excreted in the bile. So cholesterol can be directly pumped into the intestinal lumen via biliary excretion, or this cholesterol can be converted by the liver into bile acids, which can similarly undergo biliary excretion. And these bile acids actually aid in cholesterol absorption and reabsorption from the gut. So this wraps up the exogenous pathway. So in contrast, for the endogenous pathway, we are actually going to begin here in the liver, where triglycerides and cholesterol is either supplied by the exogenous pathway or synthesized directly. Triglycerides and cholesterols are then packaged with phospholipids and apoproteins to form VLDL, or very low density lipoproteins, which are kind of like the chylomicron of the liver. So VLDL is going to similarly travel through the circulation, where triglycerides are going to be hydrolyzed by lipoprotein lipase, or LPL, in the capillary lumens. And this is going to liberate free fatty acids to the muscle and adipose tissue to be either utilized for energy or stored. This is again going to shift the content from being very triglyceride rich in VLDL to IDL, or intermediate density lipoproteins, that are very cholesterol rich. Further triglyceride hydrolysis in the circulation will eventually result in LDL, or low density lipoproteins, which have the highest cholesterol content. LDL can actually interact with LDL receptors on the liver, which will trigger its uptake and clearance from the peripheral circulation. The cholesterol in this LDL will then be repurposed similar as before. LDL receptors are also found on extrahepatic tissues, and upon LDL binding to these receptors, it's really just going to deliver cholesterol that's needed for cellular processes and homeostasis. So for any reason excess cholesterol is deposited at these extrahepatic sites, it will be transferred to high-density lipoproteins, or HDL. Now, HDL is responsible for the reverse cholesterol transport, shuttling cholesterol from these extrahepatic tissues back to the liver to then be excreted. HDL can also swap up some of this cholesterol that it's picked up with triglycerides from other lipoproteins, which are also eventually cleared by the liver. So this briefly summarizes the endogenous pathway. So in the case of hyperlipidemia, modulation of cholesterol levels alters LDL receptor expression, which impacts the extent to which the liver can clear LDL from the peripheral circulation. So if we go to our next slide. So with an increase in dietary cholesterol intake and absorption, there's a greater amount of cholesterol that's being shuttled to the liver via the chylomicron remnants. This increased cholesterol binds the transcription factor that is responsible for promoting the expression of the LDL receptor. And this sort of signals to the liver that it's in a high cholesterol state. This triggers a negative feedback mechanism that downregulates LDL receptor expression in the liver, and this reduces LDL clearance from the peripheral circulation because the liver doesn't need any of that additional cholesterol that's being carried by the LDL particles, and LDL begins to accumulate. The accumulation of LDL and its extended presence in the peripheral circulation increases its chances of interacting with reactive oxygen species and becoming oxidized LDL. Oxidized LDLs are then recognized by scavenger receptors on macrophages and taken up by these macrophages to eventually form foam cells. And these begin to deposit and accumulate until they begin to form a fatty streak, eventually an atherosclerotic lesion, and potentially an occlusion resulting in the form of a stroke or MI. So if we go to our next slide. So in order to counteract these effects, therapeutics are generally targeted at augmenting LDL receptor expression on the liver or altering triglyceride and cholesterol transport throughout the body. We will first go over the statin agents, which fall under the category of augmenting LDL receptor expression, and these are indicated first line. So if we go to the next slide. So as you know, de novo cholesterol synthesis in the liver requires the conversion of HMG-CoA to mevalonate or mevalonic acid. And this is the rate-limiting step in cholesterol synthesis and is carried out by HMG-CoA reductase. Statins mimic the transition molecule of this reaction and outcompetes the endogenous substrate for these enzyme binding sites. And this inhibits this step in cholesterol synthesis. And this ultimately signals to the liver that it is in a low cholesterol state. This low cholesterol state triggers an upregulation of LDL receptor expression in the liver. It also downregulates LDL receptor degradation. The result is more LDL receptors are available to promote LDL clearance from the peripheral circulation, ultimately significantly reducing LDL levels. In addition, it's thought that statins reduce the LDL secretion because of this low cholesterol signal in an attempt to hold on to the cholesterol that it currently has. This leads to reduced triglyceride levels because of this reduced VLDL in the circulation. Some other effects that are attributed to statins, like elevated HDL levels, improved endothelial function, increased plaque stability, and reduced platelet aggregation, are also incredibly beneficial in the bigger clinical picture, but are much less understood than this mechanism that I have gone over. So if we move to the next slide. So the next set of agents that are still going to fall in this category of augmenting LDL receptor expression are going to be the PCSK9 inhibitors, which include Repatha and Praluent. So if we go to the next slide. So the proprotein convertase subtilisin kexin type 9, or PCSK9, protein helps regulate the number of LDL receptors expressed in response to cholesterol levels within the cell. So if you remember in the previous slides, I mentioned that increased dietary cholesterol delivery to the liver downregulates LDL receptor expression. Well, PCSK9 is responsible for binding the extracellular region of the LDL receptor. And if present, upon LDL binding its receptor, the entire complex will be internalized and eventually degraded, the receptor and the lipid. But if PCSK9 is not bound to the LDL receptor when LDL binds, the complex will be similarly internalized, but instead the LDL receptor will not be degraded, just the LDL. And this receptor will then be recycled back to the surface where it can aid in further clearance of LDL from the peripheral circulation. So these PCSK9 inhibitors are actually monoclonal antibodies that recognize PCSK9 and prevent its binding to the LDL receptor. Therefore, when again LDL binds its LDL receptor and PCSK9 is not present, then the LDL receptor will be similarly taken up by the liver, but again will not be degraded and will be recycled back to the surface where it can continue to aid in LDL clearance from the peripheral circulation. So these agents are very effective, but they are incredibly costly because they are monoclonal antibodies. So they're really only used when someone cannot take a statin or cannot reach their LDL goal with a statin. So moving on to our next set of agents on the next slide. We will talk about bile acid sequestrants or resins like colsevelam, colstipol, and colstyramine, which also augment LDL receptor expression. So moving to the next slide. So if you remember, the liver can convert cholesterol into bile acids, which are negatively charged, and these are excreted into the intestinal lumens via biliary excretion to act as a digestive surfactant to aid in the absorption of dietary lipids. So almost all bile acids are reabsorbed and recycled in this process. However, administration of bile acid sequestrants which are large, positively charged molecules, will bind these negatively charged bile acids and prevent their reabsorption. And because of the size of these bile acid sequestrants, they are not absorbed either. So the end result is this fecal excretion of these bile acid sequestrant complexes. This depletion of bile acids is sensed by the liver, which depletes its cholesterol stores in order to generate more bile acids. This triggers a low cholesterol signal in the liver, which begins to upregulate LDL receptor expression. And this ultimately leads to a significant reduction in LDL levels because of this increased LDL clearance and need to make more bile acids that are continuously being excreted via fecal elimination. So these agents are not as good at lowering LDL levels as statins, but they may be used as an add-on in some cases. Now let's move to the next slide and our last agent that falls in this group of augmenting LDL receptor expression. And that's going to be ezetimibe or Zetia. And this is a cholesterol absorption inhibitor. So Niemann-Pick C1-like 1 or NPC1L1 sterol transporters are found in the intestinal brush border. And they are responsible for facilitating the uptake of cholesterol into epithelial cells to eventually become incorporated into chylomicrons. So ezetimibe inhibits the NPC1L1 transporters, reducing cholesterol delivery to the liver. This again is sensed by the liver as a low cholesterol state and triggers the upregulation of LDL receptor expression. Just as before, this leads to an increase in LDL clearance from the peripheral circulation and a significant reduction in LDL levels. Ezetimibe is also not as effective at lowering LDL as statins, and it's generally used as an add-on, and it's actually combined with simvastatin into a single combination pill that's called Vytorin. So this overview sums up those agents that act to augment LDL receptor expression. But now on this next slide, we're going to jump into a new group of agents. The last groups of agents we're going to talk about fall under that category of altering triglyceride and cholesterol trafficking via indirect mechanisms. And we're going to start off with the fibrates on the next slide. The mechanism of action of fibrates like fenofibrate is still somewhat unclear, but we do know that these agents interact with and activate PPAR-alpha. And this is also known as peroxisome proliferator activated receptor alpha, or again, PPAR-alpha, which is a transcription factor. So PPAR-alpha is found in the liver, and once activated by these fibrates, promotes the transcription of genes associated with fatty acid oxidation. In the liver, this increase in fatty acid oxidation and beta oxidation triggers the hydrolysis of triglycerides to free fatty acids. These free fatty acids will undergo beta oxidation to become acetyl-CoA, and this depletes the triglyceride stores in the liver. This means that there is a lack of triglycerides to be packaged with cholesterol to form VLDL particles. And therefore, there's going to be a reduction in the amount of VLDL secretion into the peripheral circulation and a downstream reduction in LDL in the peripheral circulation as well. PPAR-alpha activity also reduces the expression of an apoprotein that is essential for VLDL packaging. So without this protein, this just further reduces the likelihood that VLDL is going to be secreted into the circulation. PPAR-alpha also increases lipoprotein lipase, or LPL, activity in the peripheral tissues to, again, promote the hydrolysis of triglycerides to free fatty acids. And these free fatty acids are then fed to the liver with the idea of repleting these triglyceride stores. But again, because of this increase in fatty acid oxidation, we will just continue to deplete this. So the reduction in circulating triglycerides is multifactorial. PPAR-alpha also increases the transcription of apoproteins that decorate HDL particles. So this increases the reverse transport of cholesterol out of the circulation and back to the liver for excretion. Therefore, fibrates generally increase HDL levels. And again, they are very effective at lowering triglycerides. However, their effect on LDL seems to be more variable and dependent on a number of factors, which is why they're generally used when triglycerides are a concern. So moving to the next slide for our very last agent. And our last agent that's going to alter cholesterol and triglyceride trafficking is going to be niacin or nicotinic acid or vitamin B3. So moving to the next slide. So just like we discussed with the fibrates, when triglyceride stores in the liver need to be replenished, adipose cells will hydrolyze their triglycerides to free fatty acids. And these will then be delivered to the liver to participate in triglyceride synthesis. And these triglycerides will either be stored in the liver or they will be packaged into VLDL particles. Now, niacin actually prevents this process by interacting with a GPCR. And this GPCR is found in the adipose tissue, and it is known as GPR 109A. So when niacin binds to this GPCR, it activates GI adenyl cyclase pathway, reducing cyclic AMP production. This leads to reduced lipase activity in this adipose tissue. Therefore, this triglyceride hydrolysis is prevented. Free fatty acids are not being shuttled to the liver. These triglycerides are not being made, and therefore there is a significant reduction in the amount of VLDL that's being packaged up and secreted by the liver into the peripheral circulation. And as a downstream effect, these VLDL particles cannot become LDL, and this leads to a significant reduction in triglycerides and LDL levels. HDL also will increase because niacin prevents the clearance of HDL apoproteins, so they hang around longer. But the cholesterol that is reverse transported by HDL to the liver is still being cleared from the circulation. The end result is a very favorable improvement in the entire lipid profile, but generally niacin is used when triglycerides are concerned. So moving on to the last slide for a quick summary. So remember, statins are the cornerstone of therapy, and the remaining agents for the treatment of hyperlipidemia fall into two groups. They either are primarily LDL lowering agents through LDL receptor augmentation, or triglyceride lowering agents that indirectly alter triglyceride and cholesterol trafficking. So I hope this session has helped you identify where the indicated agents augment this disease mechanism and fall into the clinical picture.